genomic and molecular biology data. And that's, right now, this is not currently routinely done. So there's a wealth of expertise here in the room. We've heard a lot. There's a lot of wonderful data, a lot of wonderful algorithms. And we encourage those folks with that expertise here to engage with this program and share their expertise as well. And what we look for is we look for these researchers that are in the field, that have access to species, have a knowledge exchange, generate SOPs, have the sequencing done in the field, maybe the data is analyzed in the cloud, and the final reporting, annotation, and such can be done in collaboration as an exchange. So we're building up this capability in the field. As we go through the presentations, I would encourage everybody to grab a snapshot of this QR code. The purpose of Slido is for us to aggregate questions. So if you're going to ask, if you have questions of the upcoming speakers, we'll be tracking them. And then at the end, we'll have a couple of options. One will be Slido, and I'll be able to focus in on the most important questions. You can upvote existing questions. We'll also have a couple of colleagues, and there's a couple of mics placed throughout the room, and we'll be able to use those as well if you're not capable of connecting to Slido. But we do encourage that as a first way. Okay. Want to take a moment to introduce our first speaker. Indivert Mandaretti is a high school student from San Jose. I had the pleasure of meeting him in person, and his father. His father's here as well. I would say he's much more than a high schooler in terms of his capabilities and accomplishments to date. His research interests include a wide variety of genomics, CRISPR, and a variety of other projects that as I mentioned. He'll be coming up to speak about his most recent project, which involves the sequencing of the sequence to find the first genome of the freshwater angelfish. We're not conducting scientific research. He can be found taking care of numerous aquariums and underwater pets in Beaver.
Testing. Okay. Thank you, Paul, for the wonderful introduction. Hello, everyone. I'm Indivert, a high school senior from San Jose, California. And today, I'm really excited to talk about my project, first ever whole genome sequencing and de novo assembly of the freshwater angelfish. So, as Paul alluded to earlier, throughout high school, I've been an avid fish keeper and aquarist, keeping a variety of fish, including koi, goldfish, clownfish, and more. One of my favorite fish are my angelfish that you can see on the screen in front of you. But unfortunately, in March of 2021, one of my many angelfish passed away. The photos you see, a fun fact about the angelfish is that it's a fish that I raised from birth. So on the left, you can see the mother fish, and behind the mother fish, you can see these white little specks, which are baby developing fish fry. And on the right, you can see one of those fry fish all grown up. And that's what I sequenced the genome of. So I wanted to pay a small tribute to my beloved pet and sequence its genome. I've been conducting research at BioCurious, a local community biology lab, for around 2 years at this point. But I had never done any genomics or bioinformatics work before this. And I set up to learn the basics of genome sequencing and genome assembly. And it was during this learning process that I learned about Oxford Nanopore and their sequencing technology. There was minimal literature on DNA extraction from angelfish, as this again was the first time it's been sequenced. So I experimented with 2 different NEB kits to extract the DNA from the angelfish tissue. The traditional NEB genomic DNA extraction kit worked pretty well. I got around 20 to 40 kilobases length DNA from it. In terms of the library preparation, I followed the standard ligation library prep that was performed in the angelfish DNA, again with the standard protocol. 2 R10 flow cells were required for this work. And one important part of this project that I think I want to point out is that I crowdfunded for the funding to purchase the flow cells and the nanopore device, over $1,000 through the experiment.com crowdfunding platform. In the end, I'd say the sequencing was successful. I got around 10 gigabases of DNA. In more information about the bioinformatics, I used a variety of open source tools, mainly the Galaxy Bioinformatics platform. I used Fly to assemble the genome, Busco to assess the completeness of the angelfish assembly, Kraken to determine potential contaminants in the genome, more on that later, and Raycon to polish the angelfish genome. For the mitochondrial DNA assembly, I used the Minot platform to essentially identify breeds that corresponded to mitochondrial sequences, and use the Shasta de novo assembler to put the mitochondrial genome together. In the end, the genome assembly was pretty successful. I had an 86.5% complete genome, 15,000 contigs, around 700 megabases in size with a 41% GC content, which is relatively standard for fish genomes in general. For the mitochondrial genome assembly, all 37 conserved mitochondrial genes were identified, including 2 rRNAs, 13 genes, and 22 tRNAs. And this new assembly was found to be 25 base pairs longer than the current reference mitochondrial assembly for the angelfish. For orthologous genes, I used NCBI BLAST to identify 24,000 protein coding sequences, and they were most commonly attributed to Artrocentris centrorchus, a closely related South American cichlid. So this was really interesting. In terms of cause of death, I also found that Pseudomonas aeruginosa, a common aquatic pathogen, was a common contaminant of the DNA sequencing reads. And although I can't confirm it for sure, it does possibly indicate that this bacteria was the cause of death for my pet fish. So in terms of some broader impacts and some closing remarks, this work was the first to ever sequence and assemble the genome of the freshwater angelfish. I used the DNA of my pet fish for this work, and I conducted it at BioCurious, a local community biology lab. I crowdfunded over $1,000 to cover the sequencing costs, and my genome is currently deposited to NCBI as the current reference genome for the angelfish. And I hope that one day the angelfish can be used as a potential model organism for future biomedical and biological research. I would like to thank my lab colleagues from BioCurious for their advice and guidance during the course of this project. And I want to give a big thank you to Paul and Oxford Nanopore for not only the opportunity to speak here, but for also making their lab equipment affordable and accessible to even a high school student. Thank you.
Not only the accessibility, but the ability for anyone. And Endeavor is an exceptional high school student. After speaking with him last night, but it is pretty amazing. You know, a high school student can pick up all this capability and be able to go to the entire start to finish while he's applying for college entrances, doing his coursework and that. So just, you know, a huge shout out to your capabilities, and I look forward to your successes going forward.
Thank you.
Do you have the,
Yes.
just one more maybe? Just a reminder, I think we have, just a reminder slide out. We've gotten just a question for myself, while others load up. What do you think your next steps are? Are you going to, think you're going to continue working on the angelfish, or do you think you're just looking forward to sort of undergrad?
A little bit of both, but I plan to sequence the transcriptome of the angelfish next to kind of get a better idea of the genomics of this fish. So that's next, but I'm excited to see what the future holds for me, and I want to learn everything that I can.
Fantastic. One more question that came in. People were curious about BioCurious.
Yeah.
So can you describe a little bit of what other projects go on there up in San Jose?
Yeah, so BioCurious was a community initiative founded by a bunch of citizen scientists who wanted to create a lab space where students could come and do scientific research. So a lot of the other projects are also very community oriented. There's a vegan cheese project, There's a project involving cuttlefish, where they raise cuttlefish in the aquarium at the lab. There's sequencing companies that run out of BioCurious. So you get to meet a very diverse group of people, from other student scientists to entrepreneurs at BioCurious.
One last question, and if there's any questions from the audience, if people don't, please go up to the mic, or my colleagues can bring one, just raise your hand. Great talk. Did you do the bioinformatics work on a cluster or on your own personal computer?
Right, so I use the cluster. So Galaxy is an online bioinformatics platform that's connected to a supercomputer. My computer, unfortunately, didn't have enough RAM to do the work, the complex assembly that I wanted. So yeah, a cluster.
Okay, fantastic. Okay, any questions from the audience? Yes. Good, if you can use the mic, it'd be great. Thanks.
Thank you. Sorry for the